Welcome to Kashmir Talks Biology. This is Zubair's PowerPoint Biology, and today the topic of our talk is difference between prokaryotes and eukaryotes. So, let us begin. What is a cell? A cell is the smallest unit that exhibits all of the qualities associated with the living state. As such, organisms are either unicellular, made up of one cell such as bacteria or amoebas, or multicellular, made up of many cells such as plants and animals. Scientist to remember. It was Robert Hooke who discovered cell in 1665. He observed cells in the cork. Actually, they were the cell walls of the plant cell. Cell types. Cells are divided into two different categories. One is prokaryotic and another is eukaryotic. The word pro means before and karyon means nucleus. So they are primitive type of nucleus. While as U is known as true or karyon means nucleus, eukaryotes have a well developed membrane bound nucleus. Prokaryotes. Prokaryotes are the most abundant organisms on earth and fall into two distinct groups, the bacteria or the eubacteria and the archaea or the archbacteria. Some of the main characteristics of prokaryotic cells are they have no nucleus, they have no membrane bound organelles, just ribosomes. All are unicellular. They are forerunners to eukaryotic cells. They are smaller and much simpler. The DNA is circular, and the best example we can provide of prokaryotes is the bacteria. Next, so let us talk about the prokaryotic cell structure. Each prokaryotic cell is surrounded by a plasma membrane. The cells have no subcellular organelles, only infoldings of the plasma membrane called mesosomes. The deoxyribonucleic acid or DNA is condensed within the cytosol to form a nucleoid. Bacterial cell wall. The peptidoglycan protein and oligosaccharide complex cell wall protects the prokaryotic cell from mechanical and osmotic pressure. Some antibiotics such as penicillin target enzymes involved in the synthesis of the cell wall. Gram-positive bacteria have a thick cell wall surrounding the plasma membrane, whereas gram-negative bacteria have a thinner cell wall. That is surrounded by an outer membrane in which there is a small space called periplasmic space. Some prokaryotes have a tail-like flagella. By rotating their flagella, bacteria can move through their surrounding media in response to the chemicals, a process called as chemotaxis. Bacterial flagella are made up of protein flagellin. In contrast to microtubules in case of eukaryotes, they form a long filament which is attached to the flagellar motor by a flagellar hook. Next, what are eukaryotic cells? Eukaryotic cells have a well-defined nucleus and with a nuclear envelope. They are bigger and more complex than prokaryotic cells. They have a membrane-bound organelles like Golgi complex, Endoplasmic Reticulum, lysosomes. DNA is double-stranded and forms chromosomes, that is the highly organized form of chromatin. Eukaryotes can be unicellular or multicellular. Examples are animals, plants, and fungi. Now let us talk about the eukaryotic cell. First, let us talk about the plasma membrane. The plasma membrane surrounds the cell, separating it from the external environment. It is a selectively permeable barrier due to the presence of specific transport proteins and has receptors that bind specific ligands. It's also involved in the process of exocytosis and endocytosis. Nucleus. The nucleus stores the cell's genetic information as DNA in the form of chromosomes. It is bound by a double membrane, but pores in this membrane allow molecules to move in and out of the nucleus. The nucleolus, the nucleolus within the nucleus is the site of ribosomal ribonucleic acid synthesis. Now, endoplasmic reticulum. It is the interconnected network of membrane vesicles that is divided into two distinct parts. The RER, or called as rough endoplasmic reticulum, which is studded with ribosomes, is the site of membrane and secretory protein biosynthesis and the post-translation modifications. While as the smooth endoplasmic reticulum is involved in the phospholipid biosynthesis and in the detoxification of toxic compounds. The Golgi bodies. The Golgi apparatus is a system of flattened membrane-bound sacs, popularly called as sorting and packaging center of the cell. It receives membrane vesicles from the rough endoplasmic reticulum, further modifies the proteins within them, and then packages and modifies the proteins in other vesicles, which eventually fuse with the plasma membrane or with other subcellular organisms. Mitochondria. Mitochondria have an inner and outer membrane separated by the intermembrane space. The outer membrane is more permeable than the inner membrane due to the presence of porine proteins. The inner membrane, which is folded to form cristae, is the site of oxidative phosphorylation, which produces ATP. The central matrix is the site of the fatty acid degradation and citric acid cycle. Now, let's summarize the differences between the prokaryotic and eukaryotic cells. Prokaryotic cells are small, while as eukaryotic cells are large. They are always unicellular, while as eukaryotic cells are often multicellular. No nucleus or any membrane-bound organelle is found in case of eukaryotes, while as eukaryotes always have a nucleus and membrane-bound organelles. The DNA in case of prokaryotes is circular, while as DNA is linear and associated with histone proteins to form chromatin in case of eukaryotes. Ribosomes are small, made up of 70S, while as eukaryotes have a large ribosomes made up of 80S. S refers to the Swedberg unit involved in the centrifugation. Prokaryotes don't have any cytoskeleton, while as eukaryotes have a cytoskeleton in the form of actin filaments, intermediate filaments, and microtubules. Cell division in case of bacteria takes place by binary fission. It usually takes about 20 minutes in case of E. coli, while as cell division takes place by mitosis or meiosis, depending upon whether the cells are somatic or gametic. Reproduction is always asexual in case of prokaryotes, while as reproduction is asexual or can also be sexual. So far we have talked about the differences between prokaryotes and eukaryotes. Let's conclude this session by talking about the similarities. Both prokaryotes and eukaryotes contain all the four biomolecules, that is lipids, carbohydrates, proteins, and nucleic acids. Both of them have ribosomes, although they differ in the size. Both of them have DNA, Bacteria have in the circular DNA, while as eukaryotes have a linear DNA. They have similar metabolism with minor differences. They can both be unicellular, and both of them have a plasma membrane or a cell wall. Thank you for watching. For more videos, visit us at www.facebook.kashmirtalksbiology. For more questions and suggestions, you are welcome to contact us at zubi.biotech@gmail.com. Hope you enjoy the video. Thank you very much.